Welcome to The Explainer, everyone. Today, we're diving into something absolutely wild. We are taking a journey through what is, without a doubt, the most intricate and flawless supply chain on Earth. And no, I'm not talking about Amazon or some massive global shipping network. I'm talking about the bustling, microscopic supply chain operating inside your very own cells at this exact second. We're going to explore the endomembrane system, mapping out exactly how raw biological materials are manufactured, sorted, tagged, and shipped, all just to keep you alive. Okay, here's our roadmap for today. We'll start with 1, the cell's logistics hub. Then move to 2, protein birth in the ER. 3, the Golgi post office. 4, cisternal maturation and export. 5, secretory pathways and destinations. And finally 6, when the system fails. Let's get into it. It really all starts with this staggering question. Seriously, think about this for a second. How on earth does a cell know exactly where to send its millions of newly minted proteins? I mean, every single second of every single day, your cells are churning out massive amounts of cargo. And each one of these microscopic packages has to reach a highly specific, exact location. It's a logistical nightmare. So how exactly is order maintained in all this microscopic chaos?
To figure that out, let's step right into Section 1, the cell's logistics hub.
Picture this. Right outside the nucleus, we have this massive interconnected network. First, you run into the heavily folded maze of the endoplasmic reticulum, or ER. And then, just beyond that, you've got the stacked disks of the Golgi body. Now, you really have to remember that these aren't just independent, floating bags hanging out in the cell. They represent a highly coordinated, continuous processing line. This specific geography is exactly where our journey begins.
Which brings us to the start of a protein's life cycle in Section 2, protein birth in the ER.
To really understand this manufacturing floor, we gotta contrast two very distinct regions of the ER. First up is the rough ER. It is literally studded with these protein manufacturing factories called ribosomes, which gives it this rough, granular look under a microscope. This is where proteins are actually synthesized and where they get their very first chemical tags. On the flip side, we have the smooth ER. It totally lacks ribosomes and instead forms these tubular structures dedicated entirely to making lipids. But since we're tracking a protein today, our focus is squarely on the busy manufacturing floor of the rough ER.
Look at this structure. Notice those large double membrane sheets stacked on top of each other? It basically looks like a multi-level parking garage. Well, actually scratch that. It's more like a parking garage where the ramps themselves are part of the structure, these really cool helical ramps known as taurossine ramps. When a brand new protein is formed, it enters the space inside this garage, which we call the lumen. Here, right on the loading dock, our nascent protein is folded up and gets its very first molecular tags, like a 14-sugar backbone, before it's packed into a transport vesicle and shipped out.
So once our protein leaves that ER garage, where does it go? Let's move to Section 3, the Golgi post office.
The absolutely crucial point here is understanding what the Golgi apparatus actually is. First observed way back in 1898 by Camillo Golgi, you can essentially think of it as the cell's ultimate master post office. It takes in the raw cargo from the ER, selectively modifies it, packages it, and completely labels it for its final destination. But listen, do not picture the Golgi as some boring, static sack. No way. It is a highly dynamic, bustling processing center that handles cellular packages with just mind-blowing precision.
Let's break down the actual steps of this micro-logistics journey, kind of like a masterclass. Step 1. Our protein package, which is safely enclosed in a transport vesicle coated in CoP2 proteins, buds right off from the ER. Step 2. It travels over to and fuses with the entry side of the Golgi, which is known as the cis face. Step 3. The cargo then moves into the middle compartments, the medial cisternae. This is where the real magic happens, guys. Enzymes strip away certain parts of the protein and tack on new ones. And finally, Step 4, the fully modified protein hits the exit side, the transface, where it gets boxed up for its final delivery. But how exactly does this cargo move from the cis entry door all the way out the trans exit? That's Section 4, cisternal maturation and export.
So as our protein works its way through those Golgi stacks, it undergoes a super vital process called glycosylation. This is where specific enzymes sequentially add, remove, and modify sugar monomers on the proteins. Now, why does this matter? Because these sugar modifications act as the actual zip codes written directly onto our protein packages. For example, the Golgi might slap on a specific tag called mannose 6-phosphate. That specific molecular barcode is exactly what tells the cell routing system where this package has to go. Now, our scientific understanding of this whole transit system has actually evolved quite a bit. The older view, Model 1 here, suggested the Golgi was just made of stable, fixed compartments, and the cargo had to sort of hop from one room to the next using tiny vesicle shuttles. But Model 2, cisternal maturation, completely flips the script. It suggests that the Golgi sacs, the cisternae themselves, aren't fixed rooms at all. Instead, they physically mature and move forward themselves from the cis face to the trans face. So think of it less like walking through a building and way more like standing on a dynamic conveyor belt that changes its own structure and enzymes as it carries you along. It's such an elegant solution, and for sure, it's currently the most accepted biological model.
All right, our package has matured on the conveyor belt and got its zip code. Next up, Section 5, Secretary Pathways and Destinations.
From the Trans-Golgi network, our packages are sorted and loaded into specific vesicles destined for one of three totally distinct locations. First, you've got constitutive exocytosis. These vesicles are for immediate release, straight out of the cell, like plasma B cells that are just constantly pumping out antibodies. Second, we have regulated secretion. These packages are actually stored just beneath the cell surface, just waiting for a signal before they pop. This is literally how your neurons fire neurotransmitters. And finally, lysosomal vesicles. Remember that mannose-6-phosphate tag we just talked about? That specific zip code routes digestive enzymes directly to the lysosomes, which function as the cell's waste disposal and recycling centers.
When it works, it's an absolute marvel of natural engineering. But what happens when the supply chain breaks down? Let's look at Section 6, when the system fails.
The consequences here are incredibly severe. When the rough ER fails to fold a protein correctly, that protein essentially gets trapped inside the parking garage and has to be degraded. This backlog triggers a massive cellular alarm bell known as an ER stress response. For instance, if a specific membrane channel called the CFTR protein misfolds and gets trapped in the ER, it directly causes the devastating disease cystic fibrosis. And on top of that, chronic ER stress is now seen as a critical emerging factor in neurodegenerative conditions like Alzheimer's and Huntington's, as well as diabetes. And what about our Golgi post office? Well, congenital disorders of glycosylation, or CDGs, happen when the Golgi's tagging system is fundamentally broken. Without those vital sugar zip codes, proteins just get totally lost in transit. I mean, listen to this quote. "This is so critical that a CDG is usually lethal by the age of 2, resulting in severe neurological and organ failures." It just brilliantly illustrates that these microscopic molecular steps we're talking about aren't just abstract textbook concepts. A failure in this tagging system has very real, devastating consequences for human life.
Which brings us to our final thought for today. Because despite everything we've just mapped out, we still don't have all the answers. Look at this question. What is the exact transport mechanism by which proteins travel through the Golgi? While the cisternal maturation model is definitely the most accepted right now, researchers are still fiercely debating the role of tubular connections and rapid partitioning. It honestly remains one of the great unsolved mysteries in cell biology. And really, that's what you should take away from this explainer. The cellular universe inside you is vast, it's intricate, and it still holds profound mysteries just waiting for the next generation of scientists to crack. Keep learning, and I'll catch you on the next explainer.